Now to introduce our speaker today, Terrance Wang is a fisheries PhD student at the University of Washington. His research applies quantitative methods to marine resource management, and his current focus is on trends and drivers of marine biodiversity, particularly abundance trajectories for megafauna. His graduate research repertoire also includes modeling fishing behavior and social networks of fishing communities. Prior to UW, Terrance managed data for hurricane relief with NOAA and mapping services with Apple. So Terrance will take questions as we go. And other than that, thank you everyone for coming, and a big thank you to our speaker for sharing his work with us today. So with that, Terrance.
Yeah, thanks for coming to today's Quantum. I know I'm in competition at the time with the grad student free lunch faculty interviewee, so I appreciate it. And if you leave to go for that lunch, I won't feel offended. I'll just ask you to grab my lunch order. It's a chicken bowl. Okay, so this talk is titled Any Global Biodiversity Index Misleads Policy, Exploitation Explains Finer Scale Marine Trends. And this is work done by me along with Michael Meta-Webster from NYU and Ray Hilborn here at UW. Cool. Okay, I want to start off with a very general face value narrative of the state of the world's global diversity. I think we've all seen some kind of headline that biodiversity is in some kind of crisis now for the past couple of centuries because of human-induced forces. Some indicator of biodiversity, which started off positive, has been declining. And now we're at this state where maybe we can undo our mistakes that we've made on biodiversity and optimistically look to the future by bending the curve of biodiversity. And how do we do this? How do we do this successfully? I think I'm going to borrow a page from healthcare lingo: track, diagnose, prescribe. So if I go to a doctor with a problem, say the problem is the loss of biodiversity, the doctor first asks, okay, let's collect some data, let's track some things, let's take some blood vitals, genetic history, or family stuff. And then through all this data, the doctor can diagnose the root cause of the problem. And because of that cause, the doctor can prescribe some kind of medication or solution to address the underlying issue. And I think we can apply this to biodiversity, and we need a really strong understanding of the sequential steps of track, diagnose, and prescribe in order to successfully bend the curve of biodiversity. And for the most part, this presentation will just be on that first step, track. So how are we tracking biodiversity? Well, the Living Planet Index, which is run by the World Wildlife Fund and the Zoological Society of London, every few years they push out a report called the Living Planet Report, and they do LPI, or Living Planet Index, measuring biodiversity at a global level. And they break it down into different realms: terrestrial, freshwater, marine. I'm only really qualified to talk about marine, barely, but this is the screenshot of the marine LPI. It's a pretty stark decline. It goes down from 1 to 0.44, or a 56% decline. And they have some choice pictures of a whale, a hammerhead, and a sea turtle. And before I really dive into what the LPI is all about, I kind of want to zoom out a little bit and just talk about biodiversity indices in general. What are they? Well, I think biodiversity is a pretty multi-dimensional concept. It means a lot of things to a lot of different people. But from a very biological perspective, there's around like 6 main categories. There's genetic composition, species population, species traits, etc. I'll just be talking about species populations. That's what the LPI focuses on. Either, and I mean populations as in like abundance trends. And why are these indices important? Well, we need some kind of quantifiable measure towards progress towards some goal, whether it be conservation or management. And we also need some kind of composite index that summarizes a lot of things, distills a lot of the information out there in the world. If I show up to a policy discussion with pages and pages of Excel data, that's not very helpful to them. That's not very interpretable. So that's why we need some kind of summary statistic. And who are these policymakers or managers that are pretty high level intergovernmental organizations like the Convention of Biological Diversity, the UN, especially with their Sustainable Development Goals, and the IPBES. And how are they measuring biodiversity? The LPI, like I mentioned, some other organizations like the IUCN with the Red List. And I wanted really dive deep on what kind of data that all these indices are calculated from, what are some characteristics of the global biodiversity database. And so how do we really know, accurately estimate a species trend? Well, ideally, we would want some kind of systematic survey design that goes out very consistently, unbiased, highly selective, targeting the species that you want to know. And a good example is the NOAA Bering Sea Bottom Trawl Survey, where every year they go out to the stations. The plot on the left is the spatial stratification, and it's pretty consistent and reliable, and you can come out with some good understandings of what species trends are. This plot on the right is just a time series example of male snow crab. And that's great, but this is an exception rather than the norm. I mean, a big caveat of this data set is that it's very expensive. And most of the global biodiversity data out there is opportunistically sampled. So very heterogeneous sampling effort or methods across the world. So I'm going to step through a couple of dimensions of bias with these global biodiversity databases. The first one is space. These are the figure on the left shows the amount of data that we have in the OBIS database, where the warmer color just shows where we have more data, where it tends to be more effort. And it's no surprise that, yeah, where we have good financial resources or strong scientific programs, there's good data in those orange spots around the UK or American EEZ, but there's definitely some huge gaps in the high seas, and that doesn't really mean that there's no biodiversity happening. It just means that we don't have any data because we just haven't gone out to collect. And because we're talking about oceans, it's a 3D space. There's definitely biases along the depths of the ocean. I don't have a good figure on the biases, but you can probably imagine there's more data in shallow waters than deep waters. And then the next dimension is time. So this is just the log number of records by 20-year intervals from 1800 to now. And no surprise, we just have lots of data in most recent years, probably because of scientific advancements or different societal priorities. But biases also within a dimension can exist on multiple levels. So this was at century or decadal levels, but I can zoom in and make that x-axis just a couple of decades. I pulled this example from the Pacific Bluefin Stock Assessment, and just think of these rows as just data sources, and then where there's colors or lines, points, just shows a year where we do have data. And ideally, you want this whole plot to be filled in with long, uninterrupted horizontal lines, meaning that there's data for every year from every data source, but that's just not true. I mean, some data sources are great, but a lot of them are quite patchy. They're sporadically sampled. There's a lot of interruptions and intermittent sampling, and there's a lot of white space on this plot. Another bias is taxa. We just know some species more than others. We probably know vertebrates much better than our invertebrate friends, even though it's probably because of our priorities as a society. And again, there's biases at multiple levels. Even if I just look at birds, there's probably some species within the avian class that we know more than others. Another one is survey bias. So this example is of insects. The rows are just different kinds of traps. And then the columns are different insect groups, and then efficacy or selectivity or how well that the survey method is trapping or accounting these insects, they vary wildly. And so even though you might have something very good at capturing aquatic insects with light trapping, it's not very good for, say, ants. So it's really hard to compare these time series. They're not exactly apples to apples. It really depends on what species you're talking about, what kind of survey methods you're talking about. And another dimension is quality. And I think quality means a lot of different things, but at least in this slide, I'm talking about space and time. And if we just focus on this first column, resolution means in that time series for that point, what resolution is that point aggregating or summarizing over? So in terms of space, does that point represent 1 hectare or 1 centimeter squared? And time, is that total number of observations in a minute, an hour, a day? And then Another thing is extent. So how many of those individual units do you have? So like maybe I'm counting all the individuals in 1 hectare in a giant 100 hectare extent. And same with the time, maybe I have daily observations for a year, a decade. And these histograms show or different distributions where, yeah, we tend to sample or have some preference or know better different resolutions and extent across space and time. I think this also interacts with the life history of species. So for example, if we look at walruses, they're pretty broadly distributed in the Arctic, Atlantic, and Pacific, but because they're pretty social and they show up reliably to these breeding sites and they gather together, we can get away with not having to broadly sample their giant range. We can just show up to these known rookeries or breeding sites and maybe just sample 1 hectare and say, okay, this is pretty representative of their total species abundance, just because of their life history. But for something like leopard seals, they're pretty solitary. They're homogeneously distributed across their wide range in the Antarctic. You probably can't get away with just 1 hectare sampled. You probably have to cover a wider extent, but then there comes trade-offs with extent and resolution, assuming that there's finite financial resources and human capacity. So the point of this is just to show that there's interactions in biases with quality and say taxa, but there's also interactions with biases across all the dimensions. Yeah.
Can I ask a question?
Yeah.
Can you go back 2 slides?
Yeah.
I think I don't understand this one.
Okay.
Like, is this for replicates? What does a resolution mean? Because, like, can you, like, what if you put the Bering Sea survey on there, which is for a few decades?
Yeah.
And they, they tow very, I mean, the tow distance is very short across a grid of a large space. Like how would you translate a survey like that into the x-axis on this plot?
Yeah, I would say like each spatial grid is a resolution, and then that entire footprint of the survey is the extent. That's how I see it, even though, yeah, I know what you mean, where it's just a small trawl footprint versus like the wider grid cell. So I think maybe just the trawl distance or like the trawl area surveyed would be the resolution unit in space.
So like, what's a resolution of a century?
Resolution of a century.
It's like an observation. What does that mean?
I think the way I interpret it is like, say like total number of birds counted in a century or estimated for that century.
I would have interpreted it for like the Bering Sea, the bottom left, it'd be you do it over 2 months in a year. For the extent we've done it for 5 decades. In the top left, our resolution is the quadrant, how long our toes are, and then the top right is the total area of the Bering Sea. That makes sense? Or did I get it right?
Yeah, that's how I see it too.
And so like the time resolution, so like a century, you could go out and observe something once a century for 200 years.
Yeah.
And that would be your resolution.
And say that's, and just say that's representative of that century. And then you can string together many centuries. That's your extent.
Yeah, it's really interesting plot. I was just trying to understand what it makes.
Yeah. Well, maybe in time the word is interval and duration, but I think those words are general enough.
Could you just could you repeat back the questions, because we're losing now.
Oh yeah. Sorry, online people. Okay, yeah. So I think all in all, the data slides are really just to say there are many biases that exist along many dimensions. There's many interactions among these biases. I just see it as in a very 3-dimensional visualization of the time, space, taxa. Red is where we don't have a lot of data. Green is where we do have a lot of data. There's a lot of holes, kind of like Swiss cheese, where we just don't really, these intersections, we just don't really know much data. We just have no understanding of what's going on, but you can't say that for everything. And time series they're not exactly apples to apples. They all have different methodologies, and they can answer some questions better than others. Okay, and now bring it back to Living Planet report. So how is their database like? It's 30,000 time series, 5,000 species. It's actually a pretty comprehensive database. There's been a lot of effort. I think it was only 15,000 time series like a couple of years ago. But yeah, they keep adding a lot of good data. So how does it work? This is a graphical summary of how it works. Step one, just your raw data, just like a species abundance trend. And then step two, you log it to account for multiplicative population dynamics. You just fit a simple GAM through that. Step three, you look at the sequential changes or the interannual changes, the rates of change, lambdas, and then you plot that in step four. It's all negative because this line is all going negative, and then you apply that to some kind of arbitrary starting value at a year that the LPI tends to choose 1 at year 1970. So it's a pretty straightforward approach. It makes this LPI trend looks pretty similar to the raw data. Like at first glance, I couldn't see any major flaws. And how do they blow this up to a region or a marine or a global wide LPI? I just scribbled some lines, but the black ones are just say you have more raw data trends. Step two, it's pretty similar. You fit GAMs to each individual abundance trend. Step three, you're also looking at sequential changes for each time series. And then step four is when you start aggregating all these abundance trends into one summarized rate of change. So say all your abundance trends are on the black line. For each year, you find the geometric mean, and that's your rate of change that represents all time series for that year. And then you project that onto your starting value 1 with the LPI, and you just come out with one LPI trend. But you would probably think, okay, it's a pretty straightforward, simple approach. I think that's actually a good thing to some extent, but with all this context of all the problems with biodiversity databases, are these simple methods actually addressing the complexities of the data that they're working with? I talked a lot about the data biases. I didn't really mention any of the model biases with the Living Planet Index, but there's been a lot of literature kind of poking holes at the simplicity of the Living Planet Index. And we did a little review, and I think there's kind of two main model biases. One is the geometric mean, so how they were summarizing the rates of change for every single year across many time series, and standardization. And standardization was the process of logging it and looking at the proportional relative changes, the rate, like the lambdas, given the time series. So I'll talk a little bit about the model biases. The first is the geometric mean. So we know that's a pretty sensitive summary statistic, and maybe it's okay if you have a symmetric distribution, but this paper showed that that's not the case. These are the rates of change for, I think, 10,000 time series or so, and you see a slight negative bump there, and that's dragging that geometric mean towards the left. And the paper's point is, well, is it really representative of a small subset of species that are heavily declining, are dragging this LPI, but that's not very representative of all the species or time series in the Living Planet Index.
That's a little problematic in that the orange one, which is strong non-significant decline, has a bunch of points above 0, which would be increases.
Yeah, I think this is over the course of their entire like year range. So maybe like it goes up and down.
Is like between 2 years or something.
Yeah. I don't think this is one rate of change for all, for one rate of change per time series. I think the time series can have several rates of change. Yeah. Another one is standardization bias, and it's necessary to kind of make these time series apples to apples because the units can vary differently because of methodology. Maybe it's CPUE or density or individuals, and also some species just have different abundances. You know, it's not like a rare endangered species versus like, I don't know, pigeons. So we need some kind of fungibility in order to compare these time series. And that's changes of relative abundance, not absolute abundance. And the Living Planet Index does make it pretty clear that that's what the index is, changes of relative, not absolute, but that's not very intuitive to kind of a general audience, and it leads to some weird patterns. So take, for example, the table on the lower left, you have two populations that both starting off at 100. One increases by 10 times to 1,000, the other one decreases by 10 times to 10. That's 200 to 1,010, 405% change. But in the Living Planet world, where it's multiplicative, the times 10 divided by 10 cancels out. And so that your Living Planet Index has no change, even though the total absolute abundance has changed. Another issue with the standardization is that it tends to lean or tends to calculate more negative rates of change than positive. An example is, say I'm going out every year to do a bald eagle counts at like a local city park, and the mean population abundance is 3. But because of sampling error, maybe this year I count 5, like 2 above 3, and then this year I count 2 below 3, 1. But the absolute value of log 5/3 is less than the absolute value of log 1/3. And so that average leads to a declining LPI, one that goes from 1 to 0.75. And this problem is exacerbated with especially short time series. And when I mean short, I just mean with few years of observation, and then also exacerbated with low count time series. So time series that are, I think, especially like 10 mean abundance or below. So given that we know all these biases, this project's goal is to de-bias the Living Planet Index. And I use the word de-bias, it's kind of a statistical faux pas. It's not a real word. I think treating for biases is how we do it, but I think de-bias can be a little more understandable to a general audience. And then having improved the Living Planet Index methods, report out the abundance trends of major taxonomic groups, and then think about this in a policy context. How is this actually helpful at those discussions? So how do we de-bias the Living Planet Index? We thought of 3 interventions to target the 5 biases. The first intervention is a geometric median, and that doesn't actually exist. This is another statistical faux pas. I'm just saying finding the median in log space instead of finding the mean in log space, like the LPI does. The second intervention is curating the data, and that will help address the quality data bias and the standardization bias and quality bias. And then finally, the third intervention is to disaggregate the LPI. Yeah, Trevor?
I'm just really curious about the geometric mean issue, because I understand if you're dealing with numbers of individuals, like you have 3 populations, they're also 100 individuals, and you're asking how many individuals are there at the end of 10 years or whatever, that makes sense to just look at the individuals. But when you're asking what is the average rate of change, and you're doing a like one number divided by another number, n divided by star, you're dealing with ratios.
Yeah.
And so if you're asking what is the average rate of change, you really do need to use geometric means for that, because then like you go up, one goes up tenfold, one goes down tenfold, the averages stay the same. So the percent change you have to do when the ratios, you do have to do geometric means. So because we had this thing when we were doing the global fisheries stock assessment thing, we're trying to say what is the V of a BMSY on average.
Yeah.
And you could have 10 stocks that went down to 0.8 BMSY, and 1 stock that went up to 10 BMSY. And then the average would be like 3. Yeah. Even though almost every stock went down. So you really have to use geometric means for that. And this is basically the same. So I think there is still an argument for using geometric means when you're using ratios.
Yeah.
Just to put it out there.
Yeah. I think that like the actual value of the geometric mean is something meaningful, and then maybe the geometric median isn't so much. It's just trying to find the central tendency a little bit better. So yeah, but at the scale of like many time series or populations, maybe it gets washed out, or ideally it would converge to the same thing if we had really good data. But that's a good point.
There.
Yeah.
Do you, as part of this large enterprise here, do you have access to the actual raw data that they, your step one that you had in your kind
Yeah.
of literature?
Yeah. Well, like 95% of the data, there's like confidential data.
So are they accounting for sampling bias? Are they fitting state space models here?
No.
Is that going to drag? I mean, that error is not consistent across density. Your that bias is going to be density dependent.
Yeah. It's they do have methods listed out, like what kind of code transect or survey method, but it's pretty. I think their philosophy is just hoping that it's such a large database that on average it converges to an unbiased solution. Yeah. Okay, and the third intervention is disaggregating, and by disaggregating I mean breaking down the global marine LPI into taxonomic and exploitation category groups. I didn't talk about exploitation data bias because I don't think it's actually really been covered in the literature very much, but I'll get into it in the next couple of slides. So the first intervention, what do I mean by curating data? I just mean chucking out data that probably isn't very informative. This graph shows the mean of the mean rates of change plotted along time series length. So length is just number of years in that time series. And what I mean by mean rate of change is just draw a straight line through the time series and get that slope. That's your mean rate of change. The mean of that is summarized for each time series length. And the size of the circle just shows how many time series are of, say, 2 lengths or 15 lengths. So you would think that the mean of the mean rates of change should be agnostic or independent to the time series length. But because of the standardization bias where the shorter time series tend to have just more negative rates of change calculated because of this statistical artifact, there's a weird behavior going on this left-hand side. And there's a lot of them. There's 38% of the time series are only 2 to 5 years long. And so because of this, we think, okay, let's just chuck out anything shorter than 5 years, and then let's retain the rest of these time series just because we believe them a little bit more.
I'm sorry.
Yeah.
But I mean, as an alternative, like
Yeah.
if we assume that biodiversity loss is accelerating,
Mm-hmm.
and we have a strong bias towards more recent time series,
Mm-hmm.
then those would have a real signal.
Yeah, but I think confounded by like the length of the time series, it would just be hard to tease out. And ideally, you would say you had like a long-term survey program that started in 2000, and then by year 25, you see it start decline. Hopefully, that would be picked up in one of these more trustworthy time series. Yeah, Trevor. Or Cody, sorry. Oh yeah, you can follow up. Yeah.
I mean, it would be interesting to truncate the longest time series, so the most recent period, to kind of put them on a fair plane, like, take your 15-year ones, which probably started in the 90s or something, and just kind of truncate them to the last, try to put them on equal footing.
Yeah.
The older data points, and if all of your time series, considering the most recent data points, have the same signal as the recent short time series, then I'm kind of inclined to think that it's a real signal.
Yeah. Oh, that's a good point.
That's a really good
Yeah, I
point.
like that.
You could take that one step further and say, like, so those short time series might be like 1970, 1985, 2004. So you could take a time series that had data points from those 3 years and calculate the trend from those 3 years and see if it's similar to those, because there might be some. I could imagine there being biases in things like you have a species that's very rare and very hard to collect data for.
Yeah.
And it's rare because it's declining. And so you're only able to get 3 time points of data.
Yeah.
Because the 4th time you went up, there's nothing. Or it's just really, really hard to collect the data, or whatever reason. There's a reason why you only have 3 data points for that species. And maybe that reason is also related to being a declining species or something like that. So are those data points are they a real reflection of species that are hard to collect data for are also declining more, or is it a total artifact of that? I think Cole's suggestion is really
that's a good one. I should compare that.
But the other question would be, like, are there other covariates that also align with short time series? Are there particular groups of species or time periods? Are there recent time periods? Are there areas that are hard to survey? Or, you know, are there other covariates to explain that? I think you'll get a lot more rigorous of an answer by delving deeply into that. Even though you're probably right, but it would be great to convince yourself you're right.
Yeah, because right now this is just off a hunch that the longer time series from stronger scientific programs are just more reliable. And looking at those 2-count time series, we're scratching our heads why there's so many 2-count time series. And at least I can't say much about the marine world, but when I was looking at the terrestrial data, a lot of them just came from like bird count surveys. Just 1 year you count 5 birds, the next year you come back, there's 0, and that's the end of that bird survey. And I think those, like a lot of citizen science driven stuff, and that's like still valuable information.
It could be a door effect as well. It could be that if you go to a site and look, there were 10,000 birds here, and you go back 10 years later, and there are 2,000 birds there. We should publish. And then everyone knows, versus if they went up by 1,000, you'd be like, oh, well, who cares? We went and got 2 bird numbers and they went up by 1,000. Who cares? We're not going to publish it. So that would be another thing to look at. And there's another effect. Sorry.
No, that was interesting.
But there's another really cool effect, which is that say you are interested in studying like frogs, and you're in an area that has lots of ponds. And so you go out and look and you discover that, oh, in this pond, there's a ton of frogs. We should study the frogs in this pond. There's a lot of frogs there. And you start monitoring them for 10 years. It's very likely you would have picked the pond that had a super high abundance of frogs in that year you started, purely by chance. And so on average, you expect to see declines. Whereas the other ponds, they might be going down and up and down and up. So they might start at super low levels, but now you're not monitoring. So there's a paper on that suggests there's bias from that too. So anyway, lots of
Yeah, I mean, the other issue is there's no, the precision is being washed over here. So in 2 data points, your precision is infinite. There's no observation here. You have 2 data points, you draw a line, you know that slope for sure. So we may be investing a little too much time and energy in this. The true uncertainty is going to be much more.
I see it. Yeah, because at least I think these are the standard version of one, and like those 2 ones are crazy. Cool. Yeah, that was an interesting discussion. But moving forward, we do just chuck out anything that has 5 years or less for the time series. And I wanted to see a sensitivity of including or chucking out longer and longer time series. The red is LPI with all of the data, and then the orange is the ones I chuck out with the 2 count, green chuck out with 2 or 3 counts, and so on. And we see quite a big difference. It goes from a very declining trend to maybe a stable one to a positive one, but around kind of the 4, 5, 6, 7 mark, it stabilizes around here. So we think, okay, yeah, we'll continue just with time series with at least 6 years of data and chucking out 5,000 of 13,000 time series, which is quite a lot. And then next, we're thinking about the absolute count. So we're thinking if your maximum count is 10 individuals, is that truly representative of the entire population or species abundance? Like, how reliable is that time series? And maybe it's true for like an endangered species like the vaquita, but for the most part, species are at least maybe in thousands plus. And so this is a sensitivity that's quite similar, building off of the removal of the short time series. So we just find the maximum individuals counted for time series, and then based off that max count, we chuck it out. It's not a huge sensitivity, but we think at least removing populations with a maximum count of 50 or less and moving forward from that. So that's 2,000 of 13,000 time series.
So with the red line is a time series is all zeros then?
Red line is keeping time series that have a maximum count of 0, or sorry, 0 to 25.
Oh, okay.
Yeah. Okay, and then the second intervention is the geometric median. You've seen this plot on the left before, and these are their median counterparts. And that's not too surprising. The geometric mean is just a little more stable at finding the central tendency. And so maybe the rates of change aren't as easily swayed by outliers. So that's why it ends up all being kind of the same stable trend. And this just kind of confirms, okay, the geometric median maybe has a little more credibility than the geometric mean.
So the left one there is geometric mean, and the right one is geometric median.
Yeah. Okay, and the third intervention is disaggregation. So breaking apart the LPI, the global marine LPI into different categories. How they do it is they equally weigh all the species. So in the marine world, there's like 13,000 marine fish, a couple hundred marine birds, and 100 each for marine mammals and reptiles. And even if we perfectly sampled all the species in the ocean, you would just have a marine LPI that's dominated by 90% fish. And that would kind of wash away any important key trends if there's differences in the mammals, birds, and reptiles. So we think it's a little more useful to break it down, at least first by taxonomic classes. And then we were thinking, okay, we can refine these even more. We'll keep mammals as mammals, birds as birds, reptiles into sea turtles, cartilaginous or sharks into a non-commercial and a commercial version, and then same thing for ray fin fish, and I'll get into why. The first one is marine.
There were no inverts there?
No, this is just vertebrates. The LPI is just vertebrates. Yeah, sorry. No crab LPI. Pick up my lunch order, please. Okay, so marine reptiles, this is the x-axis is the total number of species we know out there by taxonomic order. So squamata, that's all your sea snakes, and then 1 iguana. The turtles, there's 7 species of turtles, and then there's 1 or 2 marine crocodile. And then the shading, the red just means that species is not included in the LPI, and then blue is yes. And then I put the percentages of their inclusion in the LPI on the right. And with the sea snakes, we just don't, I think we sampled 1, or the LPI sampled 1. That's not really representative of all sea snakes. Sea turtles, we have data for all 7 species, and then crocodile, nothing. So I think it would be kind of misleading to say, to calculate a marine reptile LPI with mostly sea turtle data, and it'd be incorrect to extrapolate that to the many, many species of sea snakes we just don't know much about. So we just redefine marine reptile as sea turtle. We check out that 1 sea snake data. This is a similar graph with marine mammals and marine birds. We decided to keep mammals the same just because those percentages are like roughly equal enough. And if we come out with a marine mammal LPI, I would say it's relatively representative of the cetaceans, the pinnipeds, and the sirenians. And then the same thing for the marine birds. There's just a lot more taxonomic orders. There's definitely some variation, but all in all, there's general representation across the orders. The most species order is Teragiformes, and these are your small shorebirds, things that kind of live in like some kind of salty water. And then intervention number 3, we disaggregate by exploited fishes. So this example, so instead of looking at taxonomic orders, I'm just looking at whether they're commercially exploited based off fish base or FAO. For sharks, there's a lot of species that we don't commercially fish. There's like over 1,000, and we only sample 3% of them. Those are your like ratfishes or chimeras in the deep ocean. But the sharks that we do heavily target, I think it was like just shy of 100. We know we've sampled 80% of them. There's a good understanding of that. And then same thing, but the marine ray fin fish, but at a much drastic level. Yeah, what's up?
Sorry if you said this, but the blue is before or after you filter by like 10
After I filter.
After you filter.
Yeah. So definitely, there might be a few that I'm missing just because I curated it out. But the blue is probably like the most reliable time series that we think. And so non-commercial marine ray fin fish, we have 1% of these species covered. But the commercial, we have a strong understanding, and it's not a big surprise. They have high commercial value. There's a lot of money in there. We're fishing them, so that's why there's data, and that's why we have something like the RAM legacy database, which actually the LPI uses. And so that's why there's a huge bias for exploited fishes in the LPI.
Yeah, I mean, the really interesting thing about that is that all of the global sort of analyses of what marine fish status is are all based on either things that have stock assessments for, which are all only commercial species, or things we have catch data for, which are pretty much only commercial species.
Yeah.
So all of those other ones there are not represented in any of these global analysis of how marine fish species are doing, whether they use just catch status strength, which is unreliable, or whether they use active stock assessments. They're not representative of the whole ocean.
Yeah.
Which is something that, you know, is kind of interesting.
Yeah.
And a lot of those non-commercial species, I mean, how depleted are they if we're not catching any of them? What is, you know, I mean, my guess would be on average, those ones are pretty better than things we're catching.
Yeah. Like it'd be wrong to say like, this is definitely declining because we're exploiting them. But then does that mean do we fill in this red bar with a declining trend too? Probably
not.
Probably not. Yeah. I'll get into that a little more in I think next couple slides.
We actually finished the talk.
I think I'm doing okay. Yeah, I am. That's fine. So yeah, this is figure 1. We're just plotting the mean rates of change for each species. And these numbers don't matter, but I put the mean and median and the percent of species that are positive. The red line is just a 0 reference line. And the top panel is all the marine species that we have data for in the LPI, and 51% of those species are increasing. So it's a pretty symmetric distribution centered around 0. And then these are our disaggregated taxonomic groups. And there's definitely some patterns like the cartilaginous commercially exploited short species, only 40% of those are increasing. Same with ray fin fish that are commercially exploited, 49% of species are increasing, but the rest have most of the species have positive trends for the other groups. And so all taxa groups are increasing except for the exploited fishes.
And then can you remind me, these, sorry,
Yeah.
The rates of change are from the slope fitted to the data over time, or they are from a yearly rate of change that has been averaged?
Yearly rate of change that is average. Yeah.
And so all the species are also increasing?
Yes, all 7 species are increasing, which I think is not entirely true for the leatherback. Yeah.
Sorry if I missed it. So what did you do if you had multiple time series for one species?
Multiple time series for one species, it gets aggregated. It's like kind of a hierarchical process. So like within a species, you have multiple time series. You do the geometric mean for every year, and then you have many species. Then you do the geometric mean of all the species for that year that you have data for.
Okay, but then when you curate the data, you are curating by individual time series and not by time series across species.
Yeah.
Or within the species.
Yeah, but then I still use their LPI package to do their like species hierarchy stuff. Yeah, it's kind of thorny. I didn't really get into it just because it's like a lot to explain for such little value. But does that help? Cool. Okay, and then so how do these translate into the actual LPIs? So this is the all marine LPI trend with 994 species. I should have made all these y-axes constant, so really pay attention to the values. It goes from 1 down to like 0.94 maybe, and it goes back to 1. So like a pretty stable trend. But there's definitely key differences among the taxonomic groups. So exploited sharks, that's a pretty strong decline from 1 down to 0.3, so like a 70% decline. And this is kind of in line with published literature out there. We're not the first to find this. And I think they also estimated like a 60-70% decline. So we're in the same ballpark. Cartilaginous non-exploited fish, that's a pretty stable trend line, but that's 29 species out of the 1,100 species that we know are out there. So is that really representative? Probably not. Marine birds, that's a pretty strong increase from 1 to 1.5. I think this conflicts with other literature saying seabirds have been declining, but I think there's an important distinction to make between like the definition. So we say marine birds, and that includes all those medium-sized, small shorebirds, the pterygiformes. But when we talk about seabirds, I think we're thinking about like nets and albatrosses and things that like really live out in the open ocean. So maybe the inclusion of those pterygiformes, which tend to be more positive, drag that LPI up. And then we have marine mammals, 1 to 3.5. That's a huge increase, and that's nothing too unexpected. Commercial ray fin fish, declined out of 0.65 with a modest recovery, and a pretty good sample of 459 species up to around 700 exploited fishes. And then kind of same story with these non-commercial ray fin fishes. No trend line, but 176 species sampled out of 14,000. Is that really representative? I would take that trend with a grain of salt. And then sea turtles are increasing quite well. Okay, and the point of this slide is say all trend methods converge to the same trend if given enough data. So this figure on the right is Ray's paper. Ignore the blue and purple, the purple and green line, just look at the orange line. That's kind of also like a relative B over BMSY, like a relative change in your biomass. I mean, this orange trend looks really similar to this trend, probably because we're using almost the same data from the RAM legacy database. So I think the LPI it's pretty good if you have good data, but I think this is the only category where we have good data, and there's already methods out there like this. This is a state-space model that does a little bit of a better job, I think, at capturing the overall global trend of exploited ray fin fishes. Okay, and then this figure is just showing how sensitive the global biodiversity index is, depending on what kind of interventions we choose. And so I didn't really talk about different weighting methods. That's what the LPI does. It's not worth it to get into it, but just know that they exist. And depending on how you weight the rates of change, whether you use the mean or the median, whether you check out low quality data, you can change that biodiversity narrative to be something very pessimistic to optimistic to no change at all.
Terrance,
Yeah.
I wish you didn't, but what's the actual LPI?
The actual, this is the official. The black one is the official. Yeah, and then I guess the green one is the one where we have the most interventions. But even so that green one is identical to this one on the upper left. Yeah. So yeah, I think the main point of this presentation is to say, I don't think we have the data to support any accurate estimation of a global biodiversity index. And because of all those biases, there's a lot of different ways to address these biases, but depending on how you even choose those interventions can really change that trend line. And on top of that, there's so many interactions with these biases. Like I only targeted 5 biases, but there's a long list of other ones that I didn't get to, like time and space. And it's just too thorny of a problem for, I don't know how useful or interpretable a global biodiversity index is in terms of like a very actionable policy setting. And even with perfect data, because 90% of the species out there in the ocean are fish, that just masks the pretty key differences in other species like marine mammals or sea turtles, which arguably all of these major taxonomic groups will fundamentally require different policy actions to get them where we want to be. And each of them also had different management goals, right? We don't want exploited fish to always be increasing. We want to generally shoot for BMSY, but maybe that's not the same for like seabirds where we don't exploit them. And so we need to align these global indicators to something more fit with the policy structure. And so this presentation is not really to say there is no, I'm not denying that there's global biodiversity issues, that we seriously do see a lot of bad things, but like this figure here from a couple slides ago, it does show 50% of the species are declining. But I think the better question, or what the LPI is asking, is what trend is dominating? And I think the better question is to ask, okay, what's causing the declines and increases of these species? Kind of taking it back to the track, diagnose, and prescribe. I think we have enough data for the tracking part to some degree, but I think we're a little too hung up on creating some kind of composite index. And I think what we should be looking at is diagnosing these problems and hopefully learning lessons from like successful species and applying that to the declining species, and thinking about management actions that can actually bend the curve of biodiversity. So yeah, that's it. Thank you. Yeah, Howie.
Thanks, Terrance. This is really cool. It's really hard to do. It's really cool stuff. You had a slide about your curation step of the data sets and of the time series, and there was one about space. Yeah, this one. Sorry, about the counts. So the one with the Vakira.
Okay.
So how? Maybe you said it and I missed it. You said that you're basically like those different lines you're cutting out. 25, 50, like that's the cutoff, right?
Yeah.
But like, how does the sample, like the size of your sampling area, factor into that? Because like presumably, if you're sampling, you know, a 10-meter square, you can count like 5 animals. If you're sampling 10 hectares. So like, does space factor, or like effort, I suppose?
Yeah.
That? Yeah,
that's a good point. Like if you have a very small resolution unit, like in time or space, maybe you only are supposed to count 1 deer in this like table size plot. We thought about it, but decided it was too much effort because I don't think they have a good like data column on like effort or like unit or resolution.
So it doesn't, like the data doesn't come with an indication of, I mean, I say CPU, I guess, but like, you know what I mean? Like,
not so much, but I do think these time series are generally have large extents, like it's still aggregated over, I want to say like some moderately sized locale. Like it's not down to like individual coordinates. I think there's definitely a few, but not a huge proportion.
Because I mean, I was wondering, you know, like you're throwing out good information that would give you both in this step and the previous step. Like the number of years, and I guess the 2 data points, you know, we can discuss whether or not that's a good idea to have them. But I was wondering, like, could an alternative approach to discarding time series, has anybody tried like putting weights on them depending on how long they are or like how many animals it had and that kind of stuff?
Yeah, that's a good point. We thought about weighting it. And I think we were just a little tentative on adding another like model bias or some kind of arbitrary decision. Like, how do you weight? Like even just equally weighting species, that's kind of arbitrary. And then on top of that, weighting based off, yeah. But that I think it's worth exploring. Yeah, cool.
You show the histogram of the
Yeah,
kind of fireworks. I
yeah.
think for me, like the interesting thought experiment is like that top one. Like, what did that look like before civilization, for instance? Like, what's like a null distribution?
Yeah.
And I mean, I think that's the big question. Is like, is that okay?
Yeah.
So I'm wondering if, under the hypothesis that biodiversity is accelerating, loss is accelerating. Like, what if you just truncated all these time series to like 1990, redid plot? Like, is that gonna just contract in the tails, or is that gonna shift to the right or shift to the left? I feel like there's a lot you could do with parsing your data and like looking at different subsets of it to kind of try to get at that. Obviously, the ideal if we had great time series and like starting in the 1800s, we could do it, but I feel like 1970s is probably better than 2020.
Yeah.
It gets you closer to what that baseline is.
Yeah.
Because there is like a normal rate of loss. Right?
Yeah. I'm thinking like, I mean, in like an undisturbed vacuum of a world, you would probably think everything's in some kind of equilibrium. So yeah, it'd be centered around zero, but there's definitely some variation. But I like that idea of making a histogram for each like decade, or at least like one representative or baseline versus now or in the past.
So if you go to the panel with the ribbons on it, like thinking as a stock assistant scientist, what we would do is we would do like a retrospective where we just leave off a year, replot that. What you want to see is that it's like consistent. It doesn't matter if you truncate back in time, like the trend is kind of the same. If you have really strong pattern, like some of our stock assessments, then you know that probably the answer you have today is also wrong. So that could be an interesting sensitivity that gets at that as well.
Yeah, I like that. Yeah, I think it's hard because I want to say the LPI does only have good data starting in 1980, and my guess is I think a lot of the threats already have happened, or like the biggest declines were, I'm gonna guess, like maybe 1900s to like the 1970s, and now there's still threats, but maybe at a lower rate. So trying to find, hopefully they just have good data on like a baseline or reference point.
It's a hard problem for sure. That's great stuff.
Yeah, Lichi.
I have more of an opinion question,
Yeah.
I guess, related to policy implications. Like, what do you think the management objective would be? Do you think it is to keep everything stable? Do you think it's to reduce the rate of decline of, like, given an LPI, like what would management be going towards?
I think they want it as high as possible. Like, I think whether I
like to bring it back to 1970 levels, to increase it,
Yeah, let me go to the first, like this figure wants this to just exponentially go right as high as possible. Like, I don't think that's realistic. And like just looking at abundances, like of course, like if everything recovered well, it would still hit carrying capacity, so it's still stabilized at some point. So I think you need to pair these abundance trends with like, I don't know, like density dependent indicators or something. Yeah.
So what do you think would be a more realistic goal?
A realistic goal, I do think getting it to like a reference year, kind of like maybe back to 1970, back to an LPI value of 1. But
that was a low point.
yeah, okay, maybe a little bit earlier, but then if we do earlier, I guess we don't even have data for that. Yeah, I don't. my opinion is I think things should still be increasing to a certain point, but that goalpost is very vague. Like I don't have an opinion on it. I don't even think some of the policy members know exactly what that number is. It's just I want that slope to be positive. Yeah, Trevor.
I really like Cole's comment about the histogram.
Uh-huh.
The book 51. So if we go back to that one.
Yeah.
Sorry.
No.
I mean, so is that that's mean rate of change per year, or that's just mean rate of change across the whole time series?
Across the whole time series.
Okay. So I mean, if this was a natural system, there would be natural fluctuations in population levels. The longer your time series was, the more that number would be in the middle, because over a long period of time, the average change is probably going to be very small.
And we saw actually a lot of 2-count time series just at the tail end.
Yeah, which kind of makes sense. But also because this is change over the observed time period, there wouldn't be as many in the big increased part, because a lot of those are things like seabirds, turtles, whales, things that were depleted very strongly and have since recovered very strongly. Whereas the ones on the left-hand side, presumably a lot of those out in the tail are things that we humans have done bad stuff to. That isn't just natural variability. That the ones in the tails on the left, the ones in the tails on the right are probably recovered from bad stuff happening to them. Ones in the tails on the left are probably bad stuff happening to them now. And probably would be much more extreme than you'd expect from natural fluctuations. I mean, this is kind of like the classic, if your target is BMSY, how many, and you classify something as overfished if it's less than BMSY, then half the world's fisheries would be classified as overfished because there's natural fluctuations. That was a comment, not disguise this question.
Yeah. I like that. Yeah, I like that. I mean, kind of similar to Cole's thing. It's like just like geome color, like this, just fill in by like decade or something. Yeah. CJ.
Yeah, to follow up on the data curation question from a couple minutes ago, is it fair to say that the resolution, the temporal resolution in your species is related to the species life history? So you have smaller resolution and you have faster lived species.
Yeah, they have. I think their philosophy is like they just trust that the data set submitted to them has like those collectors or scientists have already thought about it. But at least in the LPI, everything's like summarized by down to year. Nothing different from that. So 1990, maybe 1993. But I don't. Yeah, I'm gonna guess that not much thought has been put into like those year intervals and then like how fast or slow growing they are.
Sure. Yeah, I just, you curate based on like the duration, right? And I'm wondering if you should curate on like a ratio of resolution to duration. Because 2 years for sardine is bigger,
Yeah, that's a good point. Yeah, maybe like a taxonomic class specific curation strategy.
Yeah, you have lifespan.
I'm sure I can get it from the IUCN. Yeah, for 1,000 species. Yeah, I'm sure there's some kind of database out there. Yeah, cool.
Sorry.
No, you're good.
Time. Since it's a quant sim,
Yeah.
and with powerful computers and tools now, have you thought about just fitting like a huge multi-level hierarchical model?
Yes and no. So I have other chapters on doing the state space models on like seals and then sea turtles, and I'm trying to like build up towards an LPI situation. Yeah. It's a lot of computation time for just even the seals. So, but hopefully I'll get better. Okay, well, yeah, I think we are over time. Thanks for staying late. I appreciate it. Yeah.
And then next week we have Keenan Guntz talking about predicting drought and insect-induced forest mortality with deep learning. But yeah, a bit of a shift from today. So, thank you, everyone.